So good morning. Alan asked me to do a few things today. He wanted me to start by giving kind of an overview for the non-geneticists in the room about sort of where we've been with regard to autism genetics. I'm going to talk a little bit about recent progress with regard to whole genome sequencing. And then I hope that I'm able at the end to just spend a minute or two thinking conceptually about sort of the last part of this, where are we headed? This is really a story. Actually, I have to say I relish the opportunity. This is my 10th year doing this, and the opportunity to take a step back for a moment and think about kind of where we've come over that time was really lovely. And one of the things I wanted to point out is that this is really a story about moving the field, trying to identify genes for idiopathic kind of garden variety autism from really abject failure to reliable, systematic, successful gene discovery. And I think, you know, the success actually now led by the Simon Simplex collection over really a decade has been, I think, so remarkable that sometimes we forget that earlier period, sort of the bad old days of psychiatric genetics. So I want to tell you about this transition, and really it is, in many respects, a consequence of the investment that Jim and Marilyn made, Jerry Fishbach, Kathy Lord leading the development of the Simon's cohort. But the initial observation really was Mike Wigler and Jonathan Sabat, published in 2007 in Science, in which they identified that there was an overrepresentation of de novo copy number variation in simplex families. They asked specifically the question, what is the contribution of de novo mutation to simplex families versus multiplex families versus controls? And they got a pretty clear answer, and what was remarkable is it was a small number of patients. So immediately that suggested to you that if this was correct, that the effect size for de novo mutation would have to be very, very large to be able to see that in 200 families. That initial observation really directly served as the inspiration for the development of the Simon Simplex collection to put together a sample that would enrich for de novo mutation and allow, one, to retest the hypothesis, and then two, to leverage that for gene discovery. And I'm going to jump through. We got a decade to do in about 2 more minutes, so I'll move quickly. But that first effort at replication, there was a Simon Simplex Genomics Consortium, about 12 of us, which I led. Mike Wigler was also working from the same data set, looking again at copy number variation now in the Simon Simplex collection. And then first series of publications in 2011, followed by publications in 2014, that really showed spot on that there was a replication of this increased representation of de novo copy number variation, particularly in the genic portions of the genome, in cases versus their unaffected siblings. Technology marched forward. Simon's was prescient, moved very quickly as soon as the technology was available to do whole exome sequencing, and then asked the exact same questions. There are overrepresentation of de novo mutation, looking at the coding portion of the genome at single base resolution, and the answer again was a resounding yes. So a series of papers were published essentially simultaneously, my lab, Mark Daly's lab, Evan Eichler's lab, Mike Wigler's lab, all showing that there was an overrepresentation, particularly of damaging de novo point mutations in the coding portion of the genome. And again, this is from a paper, Stephen Sanders was the first author, 200 families. The idea that you would get a resounding signal from 200 families in a genetic study really, again, points to the fact that we were looking at very large effect, potentially, mutations. And then below is sort of the follow-up of that with the entire sample. Simon's sample, again, a collaboration between Evan, Mike, and my group, showing this overrepresentation. Now, there's several really important things about de novo mutation. I'm just going to highlight a couple here, but really the reason that there was such a striking contribution to gene discovery. So the first is the signal to noise property of de novo mutation is phenomenal. Right? So you have the coding portion of the genome, 20,000 genes. Essentially, there's one shot per generation for a missense mutation in the coding portion of the genome per generation. So when you're looking in the coding portion of the genome, that's it that you will see in typically developing individuals. So you can imagine that these kind of properties are phenomenal if you're looking for an overrepresentation, right? Very easy to pick out what you're interested in, and easy to begin to see whether or not there's a difference between cases and controls. That property, though, also gives tremendous statistical power, because as you think about it, again, you have a big target, you got one roll of the die per generation, per exome, right? So thinking about how likely is it that you're going to see an accumulation in any given gene, any particular spot in the genome, becomes a very powerful statistical question to be able to separate, not just as a large group, is there an excess representation of de novo mutations writ large, but can we get to single gene resolution? And the answer was yes. So I do want to say then that the development of the identification of de novo mutation, the use of technology, the creation of Simon simplex collection, together really set the stage for remarkable gene discovery. This ability or this focus on trying to define rigorous statistical methods to really be able to separate what is a chance observation in the genome from something that is truly associated really is a critical point. I'm going to come back to it in a bit. So this is just a representation in 2015, a snapshot of a culmination of then years of work, again, from multiple laboratories. And I just want to point out, I use the diagrams and figures from my own lab and our group or our consortia. But really, one of the most striking things is that the prior era in psychiatric genetics, almost you never saw replication. And then so to see multiple groups all hitting the finish line at essentially the same time, often in this case with non-overlapping samples, showing essentially the same thing, really represents a profound change in psychiatric genetics, really led by the SSC and autism genetics. The other point that I want to make about this is just summarizing both the Simon simplex collection and the Autism Sequencing Consortium data, is that we'd hope to find genes of large effect, and this group sort of on average carry very large effects when you compare it to the kinds of things that we see for other complex disorders where we're looking at common variation. So as a group, about 20-fold increase in risk. And that's a distribution of risk, so the top genes, SCN2A, CHD8, very, very large risks. So I want to tell you now about sort of now pushing to the next level in technology, which is to do whole genome sequencing. I am representing for 3 groups funded by Safari looking at whole genome sequencing. I really am sort of the last person from this group that should be presenting this data. I ask these guys if I get anything wrong not to shake their heads, but if they're deep questions, we've got a lot of the group sitting in the audience today. What I want to do, though, I made that point about the importance of really thinking deeply about how to separate chance events from true association. And really the shift that I described for you with regard to de novo mutation really also represented kind of a major shift in thinking about how to conduct large-scale genetic studies in idiopathic complex genetic disorders. So the prior era really rested by and large on candidate gene association studies that are characterized by biologically driven hypotheses and then looking for nominal significance largely, okay? And the significant shift in terms of the structure of the de novo, early de novo, and actually all the de novo work was to switch to a hypothesis-free approach and to account for genome-wide comparisons. And what essentially the take-home message is that this is bad and that is good, right? And that we should not do this. That's why I put, I learned about cognitive control, and we should do that. All right. So this was, I think, among our group, all of us have been involved for a long time in psychiatric genetics, and this is a profoundly important lesson for us in thinking about moving from whole exome to whole genome sequencing, because you're moving to a much, much larger search space. You have much less of an idea about where you're going to look in the non-coding genome. And so the way that we approached it really was to try to leverage the things that were successful previously in thinking about study design. And one of the things that meant is that we really need to think about, as we were searching this large space that we don't understand terribly well, what are all the plausible possibilities for regions of the genome, non-coding genome, that might be enriched for de novo mutation. And we ended up just kind of with this initial shot, you can see these different functional categories, epigenetic states, genes, groups of genes where you might expect that there would be an enrichment of de novo mutation. We end up with about 66,000 combinations. And then that could be reduced to about 51,000 non-redundant combinations. And then working hard with Bernie, Devlin, Catherine Rader to think about what really the independent observations or independent tests were. We came up in this initial study with about 4,000 effective tests. And when you do that, and then you look in an unbiased way, without a hypothesis, and you look in the non-coding portion of the genome, this is a volcano plot with enrichment in cases moving towards the red, and our statistical threshold, the dotted line. We do not see enrichment for a particular category of non-coding variation. This is work, again, Stefan should be up here. I think the 2 talks would have been a little bit much, so they had asked me to pinch hit, and Donna Whirling in the lab led this effort, again, as part of this large consortium. That is now, the paper reporting that is in Press and Nature Genetics. The group has now moved on to a larger sample. We're up to 2,000 families. Again, at this point, we're about 55,000 annotation categories, about 6,000 effective tests. And again, if you ask in an unbiased way, without driving hypothesis, and just say, can we see a signal? We do not for any individual category in the non-coding genome. When we do a sanity check and look back at de novo loss of function variants, or we look at ASD, at de novo missense mutations, it works, right? So you can use this approach. You should be able to identify areas that are carrying risk. And really, all that this is telling us is that we're still underpowered. If you want to use an unbiased approach, genome-wide correction, hypothesis-free approaches, we're still underpowered. There is undoubtedly the case that there is going to be signal in the non-coding genome. It is literally impossible that we see what we see in the coding portion of the genome and there's nothing in the non-coding portion of the genome to be found, right? I mean, literally impossible. So there's another way to ask this question, as opposed to saying, can we identify a specific category and association? We can ask whether or not we can get an overall signal from non-coding variation in the genome. And here, again, this is largely work from Bernie and Catherine's lab and Stefan at UCSF, along with the consortium, is to use a machine learning approach with the training set of the first families, then looking in a testing set of the remaining 1,300 families and asking, is there cumulatively a signal in the non-coding region? And we see what we would expect. And in fact, we do see an enrichment that there's a predictive ability for non-coding genome. The effect size is relatively small, but we can look further and see that really where this signal is residing, this first signal is in promoters. And if you look at other characteristics, it really is this intersection of promoters and conserved sequences in the non-coding genome that begin to give us a signal. So the bottom line then from gene discovery is that there has been tremendous progress with regard to leveraging de novo mutation, both to identify genomic segments as well as individual genes. Joe Buxbaum is going to follow up. I'm really the setup with a fantastic bonanza of autism genes from an expanded set of exome sequencing. And I think, at least in our hands right now, the story with regard to whole genome sequencing is that there's undoubtedly signal there. We may be being overly conservative. I think among people who've lived through kind of complex psychiatric genetics in the Candidate Gene Association study, we'd say that we've never been hurt by being a little overly conservative about thinking about moving forward in terms of ending up with reproducible findings. I want to take the last 2 minutes just to talk a bit about where we go from here. Given the fact that now we have, I think Joe's going to give us more than 100 genes that reach a statistical threshold from exome sequencing. But what do we do from here? And I'd say that I've only got 2 minutes, so I'm really going to have to focus on kind of high-level issues. But really, this meeting, I think, has shown where do we go from here. That, as I can say, as someone who was here in early days, and I can imagine for other people, Jerry and Jim and Marilyn and Evan and Mike and others who were here at the beginning, that the idea that we would have an entire meeting that was founded on reliable, reproducible, large effect genes that we had great confidence in, and that you could see the range of neurobiological approaches from computational to systems to just developmental neurobiology. That was the point of this endeavor, and that is clearly the path forward. Conceptually, I can tell you sort of what we've been most interested in the lab, sort of post gene discovery. So prior to gene discovery, when we were spending all of our time trying to figure out how to do this, this is what we thought would happen. We'd find a gene and we'd do kind of the traditional bottom-up approach in order to be able to understand mechanism and try to get to something fundamental about pathophysiology to be able to target for treatment. But the minute that we discovered a gene, what we recognize is that this was, a tremendously simple idea about the relationship between genotype and phenotype, and that these genes they're pleiotropic. You know, we have a wide variety of behavioral outcomes, even in model systems. Sometimes you have no behavioral outcome, even though you know you've got a gene that clearly increases risk dramatically. And the fact is that actually it was embarrassing when I started thinking deeply about that initial diagram, which I'd used for so long, as sort of as a child psychiatrist. You know, it has nothing to do with brain development, right? And so when you start thinking about this, not only you know, with regard to individual genes, but in a developmental context, and the complexity of that from a spatial and temporal point of view, is that you really run into tremendous complexity. And then for us, then the question became, how do you distinguish biology from pathophysiology? That easy to identify biology, or relatively easy, when you know you've got a gene that's got a big effect. If you manipulate that, undoubtedly you're going to see something downstream of there. But the question is, and you may see many things depending upon where you look, the question is, how do you know that you've gone from there to something that's actionable, relevant for human pathophysiology? For us, where we focused on was thinking about the notion of spatial temporal convergence. The idea that we could leverage the heterogeneity, that there were so many genes now that we were identifying that all had large effect, all led to the same phenotype, that we could use that to triangulate. That if multiple genes landed us somewhere spatial temporally, that at least should give us a hypothesis about a particular point of vulnerability. And we did that by looking first at the transcriptome. This is a project now that's been going on for years, led by Jeremy Wilsey. Now he's taking it into his lab along with Nenad Sesten. And the idea was to use transcriptional profiling of developing human brain. So these are brain regions, expression data across windows of time. You can see where birth is here. We used the genes about which we had most confidence and built co-expression networks around them with the idea that that should capture something about biology of those genes. Are they interconnected? Who else do they play with? But the question we wanted to ask was not about shared biology. It was about shared pathophysiology. And there we figured that if any of those spatial temporal networks were enriched for ASD genes that we did not insert into the analysis, that should begin to tell us something about vulnerability, specifically with regard to ASD risk, as opposed to simply about seeing shared biology, say at the synapse or with chromatin modification, et cetera. So this was first published now 5 years ago, if time flies. What we found was that the strongest signal with the most robust underlying data was in prefrontal cortex. And at a period of time, so those are just obviously arbitrary numbers, between about 19 and 24 weeks. So mid-fetal cortical development, and we could look more deeply at those networks, co-expression networks, and see that they related to deep layer cortical glutamatergic neurons. Now, our lab, Dan Geschwin's lab, Joe Doherty's lab, a bunch of people were working on this simultaneously, and there has been a striking overlap. We don't have exact unanimity on where the risk resides, but there has been agreement that mid-fetal cortical glutamatergic neurons seem to play an important role. This is not a unifying theory of autism. One point, and for a particular set of genes, we've now, this is unpublished data, repeated the analysis, moving from the first set of genes now to the second set of genes, excluding the first phase 1 genes, and we get essentially the same answer. Slight shift, or I believe resolution to the temporal properties, and then when you put the entire data set together. So we're seeing a strong signal for convergence in mid-fetal cortical glutamatergic neurons, again, in deep layer. The last point that I want to make is I think there are multiple ways to think about this notion of convergence. We're certainly not the only place that's doing this. While we move forward with looking for a general signal, or actually a specific signal in non-coding genome that tells us particularly about risk, I believe that we think that there's a way to leverage the existing data already as an orthogonal approach to think about spatial temporal resolution. And the idea here is that we can look for mutations in regulatory regions proximate to established ASD risk genes. If we can show that those mutations recapitulate what we know about the mutations that are causing autism, that gives us the opportunity then to leverage exquisite, at times, spatial temporal regulation in the non-coding genome, right? So you can imagine that if we can think about the convergence of these non-coding mutations, specifically with respect to cell type and epic, that may be an important orthogonal approach to using transcriptional data. And this is work now that's ongoing with Irini Markinskov, Papa Dimitriou, and John Rubenstein at UCSF. So I'm going to end by saying, I hope, I realize I've gone a little bit long, but wanted to stress, just how critical the SSC was to move autism genetics forward. There's been a bonanza of risk genes that you're going to hear about today. We believe that at 2,000, at least in our hands, is still underpowered to find specific risk. It's undoubtedly there. It is undoubtedly of smaller scale overall cumulatively than the signal that we're seeing, certainly for loss of function mutations. Right now, it looks like it's about on the level of missense mutations, but that is likely to be, I think, winner's curse. The strongest evidence is conserved promoters. That will undoubtedly be the tip of the iceberg. And then for us, really the conceptual point, which I could do another hour on, because I think it's such an important problem in thinking about biology at this point, or where we move from here, is really trying to think about multiple ways to address this problem of differentiating biology from pathophysiological mechanisms. Thanks for your attention.